Вот, да, вроде бы она. Да, переключаются. Так. Спасибо. Я могу уже начинать? Спасибо большое. Спасибо. Ну, начинаю. Good afternoon, ladies and gentlemen. Topic of today's lecture is this lecture number 3 on immunology. Topic is immunoglobulins or antibodies, the synonyms, and humoral cell immune response development. To repeat again how processes of interaction of cells may generate cell immune response and humoral. But major topic is immunoglobulins or antibodies. Here in this first slide, you see in the lower part of the slide drawing of theoretical structures of 5 types of the immunoglobulins. Immunoglobulin G, this is a left white-shaped structure, contains 3 arms, yellow-green, yellow-blue image you see. Well, this white-shaped molecule is named monomer. The next immunoglobulin is immunoglobulin A. This is a dimeric immunoglobulin. It means consists of 2 subunits that are analogous to monomeric structure of immunoglobulin G. This immunoglobulin A, that the secretory form, protects surface of our mucous membranes in secretions, prevent absorption of microbes to surface of our epithelial cells. In the center, you see pentameric, contain 5 subunits combined by the Fc fragments of serum immunoglobulin M. That appears first in the immune response. The next, monomeric molecule consisting of yellow and green components, so-called domains. This immunoglobulin D that serves as the membrane receptor of B cells to the antigen. And right molecule that you see here of yellow domains, you see of yellow color and purple color is immunoglobulin E that play a very important role in the development of the hypersensitivity allergy reactions. So first of all, about theory of the development of immune response. The major theory that that fits for the opinion of majority of science of immunologists is the theory of the Burnett, formulated in the 1950s years, so-called clonal selection theory of synthesis of antibodies. Clonal selection theory of Michael Burnett. Clonal selection theory fits to the majority of known properties of the acquired immune response. These properties are specificity of immune response, selective characteristic of immune response, immunological memory, and other properties that are known and describe properties of immune responses. Clonal selection theory says that one of many B cells, or many B cells are shown here, you see about 10 blue B cells that have membrane receptors for different antigens. This one of these many B cells responds to the particular antigen. Antigen in the scheme is shown by the green triangles. Antigens may specifically react to the immunoglobulin receptor of only one of these B cells shown here. Other B cells have other specificity, and what this shown antigen can bind to the receptors of these other B lymphocytes and correspondingly can activate this B cell for the maturation. So B cell responds to the particular antigen begins to divide, producing the population of the identical B cells. These B cells are shown in the lower part of the slide of the figure. All these descendants of the first initial cell selected by what this antigen form the clone, and this clone, most of cells of this clone, after multiple divisions, demonstrate transformation of B cells to the plasma cells. They are last cells, shown here in the yellow square, that produce and secrete huge amounts of antibodies, what you see monomers for antibodies, that are transported to the blood and find the bacteria and bacterial antigens, neutralizing them and participating, destroying of these microbes. Some of the cells of this clone consist of descendants of the B cell that was selected for activation by the antigen specific to this cell. Majority of the cells finally would be transformed to plasma cells that will die because plasma cells can't replicate and they are short-living cells that live only 2 days. But some of them descendants of this initial B lymphocyte activated by our antigen are memory cells. These memory cells can be transformed to the plasma cells and stop their replication, remain B cells. These cells occur in the lymphoid organs and in the repeated exposition of the same antigen to the immune system of our individual, these B cells start to be selected by the antigen, but not one that's shown in the upper part of the slide, but two or really many. And they initiate formation of their clones. And all the immune organs where these B cells occur. It means memory cells are cells, long-living cells that wait for repeating appearance of antigen to repeat all this process of the activation of immune response in the future. Of course, this immune response in the future would be higher and longer than primary because in every episode of the contact with the same antigen, number of the memory cells becomes higher, higher, and higher. Here in this scheme, you see a scheme of the primary and secondary immune response developed. In the upper part, you see curves that reflect concentration of the vertical axis of the antibodies of the two classes, immunoglobulin M and immunoglobulin G. G and green curve displays total concentration of both these antibodies. What is happening on the level of cells? Initial B cell, initially in lymphoid organs, occur only few B cells capable of recognizing the certain antigen. This antigen finds these rear cells, finally contact to them, because becomes presented to B lymphocytes, but B lymphocytes by themselves also may serve as antigen presenting cell. It means they can directly recognize this antigen. After recognition, these few B cells specific to what interesting antigen interesting for us start their proliferation. It means they undergo to the serial dividings. In this dividing, first line of the B lymphocytes, descendants of this initial specific cell that was selected by antigen for replication and activation, but first line of B cells is transformed to the plasma cells that initially produce immunoglobulin M. Immunoglobulin M appears first. Peak of this immunoglobulin concentration is comparatively low, and duration of the appearance and period where this antibody immunoglobulin M may be detected in our plasma is short. It means from 5 to 15 or 20 days. It means 2, 3, 4 weeks. Other B lymphocytes that undergo to the more steps of the replication finally are also transformed to the plasma cells, or so-called plasma cells of the second line. And these second line plasma cells produce not the immunoglobulin M, but immunoglobulin G. Immunoglobulin G appears second after immunoglobulin M. Peak of this immunoglobulin occurs 3-4 weeks after initiation of the immune response. This peak is shown stained red, peak is higher and occurs longer. Green cells shown in this left part of the scheme that reflects primary immune response. What's background is yellow here, with this primary immune response. Some B lymphocytes of the clones transform to the memory cells that can mature to the B cells and occur in the B cells that accumulate in the lymphoid organs. So this is a primary immune response. What has happened when our antigen specific that we discuss again is exposed to the human or animal? Repeated contact of the immune system to the same antigen finds many, comparatively many memory cells that initiate formation of their clones. This memory cell, every of the cell replicates, and after some steps, certain steps of the dividing are transformed to the plasma cells of the first line that produce immunoglobulin M. Immunoglobulin M in the second immune response is secreted in the same amount if you compare to the primary, and the peak occurs short time. Second line plasma cells forms the immunoglobulins, antibodies of the immunoglobulin G class, a subclass. This immunoglobulin G in the secondary response has the higher level of the ECMA and occurs longer for many months and years in some cases. And what this increasing of the amount of immunoglobulin gene in the repeat immune response is a biological result of the formation of immunological memory. What about memory in the level of cells? Of course, after second immunization, memory cells also are produced, and their number is much higher than number of memory cells that appeared in the primary immune response. It means if in the future this our individual again will be exposed to the antigen, this will be third exposition of antigen, intensiveness of the immunoglobulin G synthesis would be much higher than in the primary and secondary immune response because number of the memory B cells becomes higher. It means every new exposition of our immunized individual to same antigen causes increasing of number of the memory B lymphocytes and T lymphocytes also, if we'll say about the lymphocyte fate, and they will expand to all the immune organs, mostly of the secondary immune organs, where this clonal expansion is happening. It means spreading of the descendants of the B lymphocytes reacting to the antigen in the lymph nodes, spleen, liver, and other immune organs. This is a second immune response and immunological memory. What about antibodies? The objects that really participate in the protection against foreign entities, including bacteria, viruses, and all defective injured cells. Here you see theoretical structure of the immunoglobulin G molecule. G, this is a major immunoglobulin in the secondary immune response and major immunoglobulin in the plasma of humans. Immunoglobulin G is a monomeric molecule. What here in the drawing you see these monomers. Monomeric molecule consists of two identical heavy chains that are marked by letter H. This is a red part of the scheme. This is a first heavy chain of red color. Second heavy chain shown here is drawn black. Exception for H chains, immunoglobulin monomer of the immunoglobulin G has two light chains. Light chains are shown by the green components of this scheme. It means three arms are formed. Two upper arms, right and left, consist of the fragments of H chains and light chains. Third tail of this molecule consists of two components of only heavy chains. And what these heavy chains that form this tail that's shown in the lower part of the scheme, they are parts of the immunoglobulin molecule that are responsible for the class of this immunoglobulin, type of this immunoglobulin. Heavy chains are known as the molecules of 5 types. Hammer chain, what red and black, these hammer chains, they are present in immunoglobulin G. Mu chains in the immunoglobulin M. Alpha chains in immunoglobulin A. Epsilon chain in the immunoglobulin E. Delta chain in the immunoglobulin D. It means subclass and type of antibodies depends on the unique structure of the heavy chains. Light chains of the immunoglobulins have the identical structure and they play a role in the part of the antigen binding site of the immunoglobulin. Here the scheme you see in the lower left part of the scheme, theoretical structure of immunoglobulin that is drawn simply that immunoglobulin in the previous slide. In the right part of the slide, you see how what this variable part of the antibody of immunoglobulin recognizes antigenic determinants of antigen specifically. So you see here that molecule of monomer of immunoglobulin that's shown here, or other types of immunoglobulins, if we say about monomeric structure of immunoglobulins. Well, this molecule consists of two heavy chains that are shown gray, blue, and light chains that are shown by yellow and green color. Three arms of the immunoglobulin, you see, upper arms are marked by the letter V. V means variable part of the immunoglobulin. But half of the upper arm of the immunoglobulin consists of yellow part of the light chain and dark blue part of the heavy chain. They contain pocket. What you see, this pocket that may specifically react to the antigenic determinants of the antigen specific to this immunoglobulin. Variable means that in other immunoglobulins of the same type, reacting to different antigens, internal structure of this pocket of pocket 1 and pocket 2, two variable parts, all react to different antigens. It means first molecule of the immunoglobulin may react to the antigen of the Escherichia coli. Second immunoglobulin will react to the antigen of the Salmonella typhi, for example, specifically differentiating between these antigens. So upper fragments of immunoglobulin consisting of variable parts of the immunoglobulins are named Fab fragment. Immunoglobulin G has two Fab fragments. First, the upper right arm and left in the upper left arm. Pockets of these Fab fragments that are formed between heavy and light chains in the variable part are named Fab fragments. F-A-B, Fab fragments. This monomer has two Fab fragments, right and left. Both these fragments have the identical specificity for antigen. The third arm of this immunoglobulin that's shown lower consists of the fragments of the heavy chains only. And this part is named C, or constant region of immunoglobulin E. This constant region also is named as the FC fragment. It means fragment crystallizable, FC fragment. FC fragment is responsible not to the recognizing of antibody, of antigen, but to contact to other molecules. For example, the FC fragments of the phagocytotic cells. In the right part of the slide, you see some structure of antigen that looks like a baby with a very large head that has hands and legs. What configuration, spatial configuration of these heads and legs, if it's specific to the antibody, fits to the internal configuration of the heavy chain and light chain fragments in the variable part of the immunoglobulin molecule. If complementarity of external spatial structure of the antigenic epitope of the antigen is high to the complementarity of internal configuration of the atom in the Fab fragment, in this case, this antigen would strongly specifically bind to the antibody. And the affinity of this contact would be very high. This is the strength of the interaction of antigen antibody. If configuration of the allergenic antigenic determinant doesn't fit to the configuration of the Fab fragment, binding of antigen to the antibody would be absent. Of course, occur many intermediate forms where complementarity is partial. It means in real life, after exposing of the human or animal to the antigen, antibodies synthesized as a result of immune response will have different complementarity to the antigenic determinants and different affinity of the binding. And this appearance of many antibodies that are completely or partially specific for antibody is characterized as the diversity of the possibilities of recognizing of antigen. So, there are 5 classes of immunoglobulins that are known. In the center of this slide, you see structure of the monomeric molecule. This structure fits for the previous slide. You see here that this monomer consists of the 2 heavy chains, 2 light chains, totally 4 that are associated one to other via disulfide bonds. Every heavy chain consists of 4 domains, first, second, third, and fourth. One domain is a part of the variable part of molecule. Second, third, and fourth are parts of the constant part of the immunoglobulin. Light chains, yellow, blue, also have variable domain and a constant domain. Constant domain is named C. Variable domain of light is marked as L. This molecule has 2 identical antigen binding sites, first Fab fragment and second Fab fragment, and FC fragment, third, that cannot react to the antigen. The monomeric molecule is the immunoglobulin G. What? In the top of the slide, you see IgG. No, here you see IgG of the third subtype. This immunoglobulin can serve as the membrane receptor of B lymphocytes for antigen, occurs only the soluble molecule, and is the major immunoglobulin that occur in the our blood as a result of humoral immune response. This is the most concentrated immunoglobulin. On the left, upper part on the left, you see a monomeric structure of immunoglobulin M. C fragments of this immunoglobulin M differ from the G of course, they have portions that bind monomeric immunoglobulin M to the membrane of the B lymphocyte, and monomeric immunoglobulin M serves as the antigen receptor for B cells. Lower you see pentameric structure consisting of 5 monomers of immunoglobulin M that combine and associated one to other, stick one to other in the area of the FC fragments. This is a serum immunoglobulin M that can bind to the cells by the FC fragment, but have the maximum number of Fab fragments that are identical in the structure and react to the same antigenic determinant. In the lower part of the slide, you see immunoglobulin A. This is a secretory immunoglobulin A. This immunoglobulin consists of 2 monomeric molecules of immunoglobulin A that are combined one to other, so the joining chain. Joining chain combine FC fragments here shown as the yellow ball, and in the immunoglobulin M also the yellow ball. Joining chain. Chain responsible for binding of FC fragments of the neighboring monomers. Exception for this, you see wave of the blue color. This is a secretory component, so-called, responsible for protection of immunoglobulin A in the secretions. Immunoglobulin A has the 4 antigen binding sites, may be secreted to the secretions on the surface of mucous membranes, and is protected from digestion in the secretions by the presence of this secretory component shown as the blue wave. On the right upper part of the slide, you see monomeric molecule of immunoglobulin D. This is a second immunoglobulin that serves as a membrane antigen receptor of the B lymphocytes. It means 2 types of immunoglobulins may serve as the membrane receptors of B lymphocytes for antigen. This is monomeric immunoglobulin M and the monomeric immunoglobulin D. And the last, immunoglobulin E, lower than IgD. This is a monomeric molecule of immunoglobulin E. FC fragments of these immunoglobulins may bind strongly to the immunoglobulin E FC receptors of the mast cells and basophils. And as a result, immunoglobulin E bounded to the mast cells and basophils serve as the antigen receptor for these cells. And immunoglobulin E may cause very destructive inflammatory reactions that are developed in the process of the hypersensitivity or allergy reactions. Well, this is a brief review, and now let's characterize these immunoglobulins with the more details. So, first is immunoglobulin G. Immunoglobulin G. You see structure of this monomeric molecule. Immunoglobulin G is a monomer with the molecular formula H2L2. It means 2 heavy chain, first purple and second, and 2 H, 1 gray and second gray. All the chains, they are combined via disulfide bonds. Molecular weight of this molecule is 150,000 daltons, so 150 kilodaltons. This molecule is divalent. Divalent, it means it has 2 Fab fragments, so 2 antigen binding sites, and antigen bounded to this antibody is our scheme is shown by the red star. FC fragment of this molecule is shown as the upper fragment consisting of 2 constant parts of the FC fragment. Now, all the variety of immunoglobulin G molecules is subdivided into in 4 subclasses. 4 subclasses. immunoglobulin G1, immunoglobulin G2, immunoglobulin G3, and immunoglobulin G4. Immunoglobulin 1, IG1, may be produced during the process of the development of the cellular immune response. Immunoglobulin G2, G3, and G4, they fit for humoral immune responses or T helper responses type 2. We will characterize this later. Functions of immunoglobulin G. Immunoglobulin G is a major class of the antibodies in the blood. It's producing great amount in the secondary immune response. Normal concentration of immunoglobulin G in the human blood plasma or blood serum is from 10 to about 13 milligram per milliliter. This is the highest concentration of this immunoglobulin of certain subclasses. Highest. And this is a major immunoglobulin that is secreted in the secondary immune response. Immunoglobulin G protects us against bacteria, viruses, protozoa organisms, fungi, and toxins of microbes or other types. Here you see a list of the 5 major properties of immunoglobulin G. Immunoglobulin G is the most abundant immunoglobulin in newborns because in newborns, in the blood of these babies circulate maternal immunoglobulin G that may pass through the placenta from mother organism to the organism of the fetus and later organism to the newborn. They are maternal antibodies, and in the newborns, they serve as a confirmation of the acquired passive immune response, natural passive immune response. This presence of the immunoglobulin G in the blood of the newborns depends on the second property of this immunoglobulin. This immunoglobulin G can cross the placenta to supply protection to neonates. Third property, immunoglobulin G can activate complement via its Fc part. It means immunoglobulin G may serve as the activator of the classical pathway of the complement system. You know that classical pathway of the complement system activation is the most powerful mechanism of activation of this protective system. When Fc, fragment of immunoglobulin G, they are responsible for this process after contact of immunoglobulin G to the antigen, of course. Fourth property, immunoglobulin G binds to so-called professional phagocytes via its Fc part. Therefore, immunoglobulin can opsonize. You remember that opsonization is the process of the enhancing of immune response, enhancing of phagocytotic reaction, phagocytosis, engulfment of the microbes due to the presence on the surface of these microbes, special absorbed molecules that are recognized and attract the phagocytes for this opsonized microbe. You remember that one of the proteins of the complement system may serve as the opsonin, what's attracting phagocyte for the bacteria coated by these opsonins. So, first opsonin that you could recognize, this is a FC3. So this is a C3b component of the complement system, capital C3b. This molecule is synthesized after cleaving of C3 component of the complement system to C3a and C3b fragments. C3b attached to the bacterium, this is the first opsonin. Second opsonin is this, immunoglobulin G. Immunoglobulin G binds by its Fc fragment to the FCG receptors of the macrophages, for example, and this way attracting these macrophages to the bacteria, facilitating enhancing engulfment of bacteria. And property number 5. Immunoglobulin G participates in so-called antibody-dependent cell-mediated cytotoxicity, ADCC. Antibody-dependent cell-mediated cytotoxicity. This cytotoxicity is realized by natural killer cell. You know that natural killer cell is the cell of the innate immunity. This is a lymphocyte-like cell that cannot engulf foreign materials. It means natural killer cell is not the phagocyte and can't kill some bacteria after phagocytosis via so-called intracellular killing. Natural killer cell is responsible for external killing of the bacterium or bacterium-infected human cell. So, what if in the human body appears antibody, immunoglobulin antibody against virus antigen, and if this virus antigen is exposed on the surface of virus-infected cell, or this exposition of virus antigen on the surface of virus-infected cell makes the cell target cell for natural killer. Natural killer recognizes by its Fc receptor immunoglobulin G bounded to the target cell and initiates signal for attack of killer cell against this target cell. Attack via secretion of perforin and enzymes and secreting of the initiating of the apoptosis. With this perforin enzyme attack against the target cell and additional stimulation of apoptosis are the extracellular mechanism of the killing of the virus and bacteria-infected target cells by natural killer cells. Process mediated by presence of antibody. In this slide, you see effect of complement activation. You see green bacterium, you see yellow star-like bacterial antigen on the surface. For example, this may be the O antigen of gram-negative bacterium. This antigen interacts to the specific immunoglobulin G molecules. Here you see two blue molecules. Fc fragments of these two molecules bind to the domains of the C1 component of protein of the complement system. C1 protein is activated by this antigen antibody complex and start to work as the enzyme that cleaves C2 and C4 molecules for A and B fragments. C2A component and C4B component of these molecules combine, forming C3 convertase that converts C3 for 3A and B. These molecules C3 play a role in the formation of C5 convertase. C5 convertase produces C5A and C5B. C5B attacks to the surface of this bacteria C6, 7, 8, and 9, and altogether they form tube-like structure that is the pathological pore that causes leakage from the bacteria of the water, ions, and finally lysis of this bacteria. So, this is a classical pathway of the activation of complement system. Complement system may be activated only by two immunoglobulins, immunoglobulin M and immunoglobulin G. Other not. Other can activate complement system by the alternative pathway, but not by the classical. A classical is the most powerful. Immunoglobulins of the G subtype and M subtype that you see here play a role in the immunological testing of the infectious diseases. Immunoglobulin M and immunoglobulin G, first of all, immunoglobulin G, may play a role in the immune reaction that is named precipitation. This is a sticking of the soluble antigen by antibodies. In the reaction of agglutination, this is the binding of the neighboring bacteria that are stick one to other via antibody of the immunoglobulin G type or M. Hemagglutination inhibition tests are based on the reaction of the immunoglobulins class G or M to the hemagglutinins of the viruses. Virus neutralization test is based on the interaction of the immunoglobulins to the virus antigens. Radioimmunoassay, this is a immunological test where you use radically labeled antigens or antibodies. ELISA, enzyme linked immunosorbitol, this is a widely used immunological test that is also based on the reaction of the antigen antibody. Immunofluorescence, immunoblotting, immunochromatography. Well, this is a list of the immunological tests that are realized due to reaction of the immunoglobulins, what here in the figure you see immunoglobulin M, to the antigens. What in this comment you see 3 red blood cells, and they are agglutinated. This reaction of hemagglutination. Virus neutralization test realized by immunoglobulin G. You know that viruses may attach to the surface of the host cell, inject its genome to this cell, and initiate replication. If in the blood of humans occur immunoglobulin G or immunoglobulin M, this immunoglobulin shown blue here react to the attachment proteins of the viruses and prevent attachment of virus to the cell. In this way, virus neutralization is happening. This reaction of virus neutralization may be done in the laboratory conditions, and this reaction occurs in the real life in our body as a part of our response to the infection. Toxin neutralization test by immunoglobulin G. For example, this toxin neutralization test is used in the laboratory diagnosis of the botulism. You know that botulinic toxins are specific for receptors and bind to the receptors of the nerve cells, causing relaxation of muscle cells because neurotransmitter is not released. Neutralization test is based on the use of the antibodies specific against different serotypes of the botulinic toxin. If this antibodies fit to the antigen in their specificity, they bind toxin molecules, preventing contact of this toxin to the receptors of cells, and cells remain unaffected. After that, all these small immune complexes of antigen antibody are engulfed by phagocytes and are digested. It means this is a toxin neutralization test. But you understand that in the real life, antibodies, if present in the human or animal organism, protect this host against many toxins, not only botulinic toxin, but against many toxins of bacteria, fungi, insects, for example, or snakes. Agglutination test. Agglutination test is based on the clumping of the antigens via molecules of the immunoglobulins. Here you see agglutination that may be maintained by immunoglobulin M or immunoglobulin G. In the upper part you see of the slide 3 bacterial cells that are combined one to other in the process of agglutination via simultaneous reaction to the 5 fragments of 2 different immunoglobulins M. Lower is immunoglobulin G that also may form lattice. It means may stick many bacterial cells via contact of the 5 fragments of the immunoglobulins to neighboring bacteria. These bacteria form the agglutinates that may be recognized by the eyes. Lower is the tube agglutination test. This is a vital reaction or Vidal reaction. Reaction this is directed to the finding and measuring of the concentration of the anti-salmonella antibodies. What do you see here? You see a serial of tubes, test tubes. From left to right direction, these tubes contain serial dilutions of the serum. The highest concentration of the serum would be in the left tube and the lowest in the right. You see that first, second, third tubes have the absolute transparent liquid in the tube and agglutinates on the bottom. Fourth tube, marked by the arrow, contains agglutinates, but transparency is not absolute because some bacteria agglutinate and some not. Concentration of the antibodies in this fourth tube is tighter of this reaction. All the tubes that are shown in the right part of this system, they are translucent and they don't have agglutinates on the top. Slide agglutination is shown in the upper part of the slide. You see latex agglutination. Latex particles usually stain, but in our case here stain blue, and contain they contain the surface artificially bounded to them antigens of the bacteria or viruses. For example, the such bacteria as the Neisseria meningitis. In the left part, marked by the word B, left, you see that agglutination of this latex particles not happen. Higher you see homogeneous blue color of the mixture of the bacterial contained material, for example, cerebral fine fluid, and antibodies. In the right part, you see that latex particles are agglutinated. In the illustration upper, you see that what blue agglutinates occur visible by the eye. It means that these antigens are agglutinated by antibodies that occur in the patient. This is a positive result in the left, but negative. Immunofluorescence test is also based mostly on the using of the immunoglobulins G. Here you see immunofluorescent result of the identification of the Yersinia pestis. You see capsules of these bacteria that are stained by the fluorescent dye, and when exposed to the UV light, these capsules start to emit light, visible light, because they are stained by the specific antibody. What in the lower left part you see specific antibody that is labeled by the fluorescent dye. This is a direct immunofluorescence test, what's shown in the illustration and shown in the left part of the demonstration of machining of the test. What do you see? Surface of bacteria with different antigen. One of them is triangle. If you take specific antibody against this antigen of the bacterium, antibody binds to this antigen and binds to the surface of bacteria fluorescent dye, for example, fluorescein. After that, lighting of this bacteria by the UV light makes it bacteria visible because bacteria emit light as a result of the presence of the fluorescent dye. In the right part of the slide, you see indirect immunofluorescent test. In the direct test, you search for not the antigen of bacterium, but search for antibacterial antibody in the human organism. You take known bacteria contain bacterial antigen, you mix this. So this bacterium is stick to the surface of the slide. You put droplet of the human serum containing antibodies to the surface of this bacterium. After that, wash out excess of the antibodies. After that, binds, add to the surface of the slide diagnostic secondary antibodies. They are shown orange. These antibodies are animal antibodies specific to the immunoglobulins, for example, Fc fragments of the human immunoglobulins. And the second antibodies are labeled by fluorescent dye. In this case, fluorescence of the microbe, visualization of this microbe as a fluorescent object, is possible only if in the patient's serum occur antibody that serve as the bridge between microbial antigen on the surface of bacteria and the secondary antibody that are labeled by the fluorescent dye. In this case, you see enzyme link immunosorbent assay. This is also assay that may occur in the direct and indirect form. What do you take to realize this test? You take the plastic plate, flat bottom plastic plate. This plastic is sticky for proteins that first drop to this plastic. If to this plastic plate you will drop material that you have to study, you have to examine, you may find in this material specific antigens. Where do you search antigen? Antigen in the left direct scheme of the direct immune enzyme link immunosorbent assay, you see that black diagnostic antibody labeled by enzyme, for example, collide phosphatase, may bind to the surface of the slide only if specifically will recognize interesting for us antigen what among the molecules that were dropped to this vial of the plate. After that, you add substrate. Substrate becomes cleaved by enzyme, and from the colorless state will be converted to the visible state as the dye. In the case of phosphatase, substrate becomes blue. It means if you see blue color development in the vial, it means that diagnostic antibodies find antigen. If you search for another antigen, but search for antibodies, you sensibilize surface of the bottom of the plastic by the known antigen. In this case, you search for patient's antibody. After coating of the plastic by the known antigen, you add to the system patient serum. If serum contains immunoglobulin, for example, immunoglobulin G, this immunoglobulin G binds to the antigen, stick to the surface of the plastic. After washing out of the excess of these antibodies, you add anti-human animal antibody, shown red here. These animal antibodies react to the Fc fragments of human immunoglobulins, and they are labeled by enzyme. After dropping to this system of the substrate becomes cleaved, changes the color, and you see development of the color. More intensive color, more molecules of the immunoglobulins are formed. The next immunoglobulin. Next immunoglobulin is the immunoglobulin M that may occur in the form of the serum immunoglobulin, pentameric molecule that is shown here, or in the form of the monomeric membrane associated immunoglobulin M. Now let's start from the serum immunoglobulin M, SIgM. SIgM, the pentamer, consists of 1, 2, 3, 4, 5 monomeric units. Each of them fits to the structure of the immunoglobulin G. This pentamer is stabilized by the J chain. This is joining chain that reacts to the Fc fragments of all 5 molecules of the IgM. As a result, structural formula of this immunoglobulin is 10H and 10 light and 1 G chain. This is a very heavy molecule with a molecular weight that weighs about 500,000 daltons, or 500 kilodaltons. This pentameric molecule may simultaneously react to the 10 antigenic determinants, making this molecule is more avid than other immunoglobulins. Functions of the serum immunoglobulin M. This is a major immunoglobulin in the primary immune response, and first in the any immune response, primary and secondary. Immunoglobulin is first in the development and the immune response. Concentration in the serum, normal concentration, is from 1 to 1.5 milligram per milliliter. This is important defense against bacteria and viruses, For toxins, effectiveness of this immunoglobulin is lower than effectiveness of IgG. Immunoglobulin M, serum immunoglobulin M, has highest avidity, its capacity to be attached to the surface of the microbe. Highest avidity of the immunoglobulins because its interaction with the antigen can involve 10 of its binding sites, 10, not 2, that is typical for IgG, but 10. This immunoglobulin is an efficient activator of complement system, may activate complement system by the classical pathway. Immunoglobulin M can opsonize indirectly bacteria by activating complement. It means immunoglobulin M activates very effectively complement system. As a result, appears many C3b molecules. And these 3b molecules serve the opsonins. It means immunoglobulin M not directly seeks surface of bacteria to the phagocyte, but indirectly stimulating appearance of these components of complement system, C3b. And of course, because this immunoglobulin, serum immunoglobulin N, has the highest avidity, this is the most effective among all the immunoglobulins in the agglutination, capacity to cause the clumping of the bacteria or some red blood cells or other cells. Membrane immunoglobulin M. Membrane immunoglobulin M, what's this membrane? Is a monomeric molecule and integral membrane protein that serves as the antigen receptor B lymphocytes. So this is a B lymphocyte. This immunoglobulin M may be exposed to the active stimulated B lymphocytes and arresting memory B lymphocytes. Immunoglobulin M memory has mu heavy chain. This chain anchors this immunoglobulin to the lipid membrane, cellular membrane of the B lymphocyte. Molecular formula is identical to the molecular formula of other monomers, 2 heavy, 2 light chains. This is divalent molecule. Biological role, function. A chains of these immunoglobulins first to be produced during B cell development, and peak is the primary response. First peak is the peak of immunoglobulin M. It's an antigen receptor for B lymphocytes. This is a major function. We told about this. And immunoglobulin M may be important as a receptor of the telogenese signal that lead to clonal energy and immunoglobulin tolerance. It means this immunoglobulin M presence of the B lymphocytes presence mediates reaction of this B lymphocyte to the certain antigen. It means that in the during the formation of immune system of humans in the fetus, clones of these B lymphocytes that have M immunoglobulins specific for human antigens are eliminated. First possibility, or suppressed. Suppressed, it means prevented the activation by the contact to the antigens. But in both these cases, tolerance is happening. It means impossibility of the reaction of the own B lymphocytes to the own antigens. This signal, tolerogenic signal, may look like as the contact of the immunoglobulin M on the B lymphocyte during formation of the fetus to the own antigens with the activation of the apoptosis of these B lymphocytes and elimination of these B lymphocytes, autoreactive B lymphocytes from the human organism. The next immunoglobulin is the secretory immunoglobulin A. Secretory immunoglobulin A. Secretory immunoglobulin A formula, structure of this immunoglobulin is shown in the upper right corner. This is a dimeric molecule with a structure formula 2 multiplied by H2L2. It means this dimeric molecule contains 4 heavy chains and 4 light chains and contains 4 antigen binding sites. 2 molecules of immunoglobulin A, they are combined in the complex via J or joining chain that is shown in this now drawing as a small straight line that binds one Fc fragment to other of immunoglobulin A. But exception for joining chain, this immunoglobulin has additional so-called secretory component. Also, this component may be named as polymeric Ig receptor, PIGR. This secretory component in this scheme shown as a red sinusoid-like or snake-like molecule that overlaps Fc fragments of both monomers that are combined in the dimeric molecule. This secretory component is synthesized by epithelial cells. And we understand that all the immunoglobulins are synthesized by plasma cells. Plasma cells produce this immunoglobulin. Immunoglobulins come to the basal membrane of the epithelial cells, what here. Passage through this basal membrane is this molecule pinocytosed by epithelial cell, is transported through the epithelial cells to the surface of the cell and is released to the lumen of the organ. It means initially antigens through the M cells of the epithelium goes to the antigen presenting cells and B lymphocytes that are present in the submucous lymphoid organs, Peyer's patches, for example, initiates activation of B clones cells reacting to this antigen of the bacterium in the lumen. Then B cells are transformed to the plasma cells that produce immunoglobulin A. This is polymerized to dimers and additionally is coated by the protein secondary component, secretory component secreted not by plasma cell but by epithelial cell. This is the only example when molecule of immunoglobulin has something produced by not the plasma cell, by epithelial cell. This secretory component is important to maintain passage from immunoglobulin from internal space of our tissues to the lumen, and a secondary component prevents digestion of the immunoglobulin A secretary in the lumen, and this immunoglobulin lives much longer than other immunoglobulins if they appear in the lumen of the intestinal tract, for example. Of course, molecular weight of this molecule is 400,000 dalton, and this is a molecule having 4 antigen binding sites. Function of secondary immunoglobulin A. This is the main immunoglobulin in secretions. What are secretions? Such as colostrum, milk, saliva, tears, respiratory, intestinal, and genital tract secretions. It's important in the defense against bacteria and viruses. Secondary immunoglobulin A prevents attachment of microorganisms to the mucous membranes at the external surface of these membranes. Immunoglobulin A can neither activate complement through the classical pathway nor bind well to Fc receptors of the cells, and can pass through the placenta. In this scheme, you see what is happening in the mucous membrane after attaching of the bacteria to the epithelial cells. What number one, what first step of this figure demonstrates that two toll-like receptors on the surface of epithelial cells recognize bacterial pathogen-associated molecular patterns, terminal manose, for example. After that, toll-like receptors activate MID 88 molecule. This is an adapter protein that stimulates transport of the activation signal from the surface of cell to the nucleus of the cell, activating gene that is responsible for synthesis of polymeric Ig receptor. What polymeric Ig receptor, PIGR, this is what here arrow demonstrates that what from this sequence of nucleotides is initiated transcription and translation. And finally, this polymeric Ig receptor appears on the internal surface of the cell exposed to the basal membrane. They are brown molecules, what with these state components. What they are parts of the secretory component. Epithelial cell produces these polymeric Ig receptors, and they are transformed in the future to the secretory component. So these receptors interact with the IgA molecules, facilitate dimerization of these IgA molecules, bind these IgA molecules bind to the polymeric Ig receptors. They facilitate passage of these complexes through the cytoplasm of the epithelial cell. This is a part of the scheme number 4. And after these complexes are secreted from the cells to the lumen of intestine. During this secretion, was this state component of the polymeric Ig receptor is removed, and this polymeric receptor is transformed to the secretory component of the epithelial cell. Finally, matured immunoglobulin A, dimeric, coated by the secretory receptor, find bacteria, antigens of bacteria, and bind to this bacteria and prevent binding of this bacteria to the receptors of epithelial cells, protecting these cells, preventing colonization of the surface of these cells by the microorganisms. Immunoglobulin A, of course, initially synthesized by the plasma cells and secreted to the blood, and after from the blood transported via mucous membranes to the lumen of the intestinal or respiratory system. It means in the blood occur serum IgA. Serum IgA, this is a monomeric molecule with the formula H2L2, two heavy to light fragments, molecular weight about 170,000 daltons, 170 kilodaltons. Concentration of this immunoglobulin in the serum is from 2 to 3 milligram per milliliter. It means most concentrated is immunoglobulin G from 10 to 13 milligram per milliliter. Second rank was this serum immunoglobulin A from 2 to 4 microgram per milliliter. Third is immunoglobulin G from 1 to 1.5 milligram per milliliter. Other immunoglobulins, immunoglobulin D and E, occurs in the trace amounts. IgD. IgD, this is a second membrane antigen receptor, B cell receptor of the B lymphocytes. First is monomeric immunoglobulin M, second is IgD. IgD is present on the surface of many B lymphocytes, resting or activated. Formula to heavy to light chains, molecular weight is 180 kilodalton. IgD has only one function, antigen membrane receptor of B lymphocyte. Other functions known for antigens are atypical for this immunoglobulin. Concentration of this antibody in the blood is 3,000th milligram per milliliter. This is a 1,000 times lower concentration than the concentration of the other immunoglobulins of G, A, or M. And the last one. This is immunoglobulin IgE. Here you see monomeric molecule of the IgE. IgE structure. This is a monomeric molecule with a formula to heavy to light chains. Molecular weight about 190 kilodalton. What is the interesting peculiarity of this immunoglobulin? Fc fragments of this immunoglobulin bind to the Fc E receptors of mast cells, basophils, and eosinophils. And in this way, immunoglobulin E serves as the antigen receptor for these cells, for mast cells, basophils, and eosinophils. In the contrast to immunoglobulin M monomeric or immunoglobulin D, immunoglobulin E can be synthesized by mast cells, basophils, and eosinophils that consume this antibody, the surface receptor for antigen. Immunoglobulin E is usually secreted by the plasma cells. After that, it's passage to the blood, and from the blood goes to these cells, and after contact with the cell, binds to the surface of these cells: mast cells, basophils, and eosinophils. All these three types of cells play a very important role in the development of the anaphylactic hypersensitivity reaction or anaphylactic allergy. Functions. Cell-bound immunoglobulin E. Cell-bound. It means immunoglobulin, after binding to the Fc fragments of the basophils, eosinophils, and the mast cells. Cell-bound immunoglobulin E serves as the receptor for antigen or allergen of mast cells, basophils, and eosinophils. Concentration of this immunoglobulin in the plasma is 5 millionth, 5 millionth part of the milligram per milliliter. This is the lowest concentration of immunoglobulin, trace amounts. If concentration would be 10 or 20 times higher, it means that patient has hypersensitivity disease based on the hypersensitivity reaction, for example, helminthosis. Immunoglobulin E, what's cell-bounded immunoglobulin E, participates in hot defense against helminths. How? When helminth is attached to the epithelial cells of our intestine, immunoglobulin E binds to the antigens or allergens of these helminths in the submucous membrane, and this contact causes releasing of histamine and other biologically active substances from this mast cell in the place where worm is attached to the epithelium. This part of our mucous membrane becomes damaged and disrupted, and the worm after that has no surface for the binding, becomes more detached from the mucous membrane and released from the organism with the feces. This is a reaction very, very low, destructive, that prevents strong binding of the parasite to the surface of mucous cells. Immunoglobulin E mediates anaphylactic reaction. With this reaction, in the controlled form, this is a protective against worms. But when the antigen binding site of adjacent immunoglobulin A's are cross-linked, it means if neighboring immunoglobulins attached to the surface of the mast cells are cross-linked because both molecules of half fragments react to the determinants of one antigen molecule, or one antigen complex. Cross-linking of these immunoglobulins attached to the surface of the cells is happening. This cross-linking generates a signal for degranulation of mast cells and basophils with the release of the histamine from the vesicles to the space, extracellular space. And histamine causes changing of the vesicle permeability, intensify blood circulation in the capillaries, causes passage of the water from the vessels to the tissues, causes in this way swelling, causes increasing of the temperature, causes influx of the eosinophils to this area. It means what now may list symptoms of the anaphylactic reaction. Immunoglobulin E doesn't fix complement, so the classical pathway, and doesn't cross the placenta. You understand? If such the protein immunoglobulin will pass the placenta of the fetus will have a prevalent chance to be killed by the anaphylactic reaction in the organism of the mother before delivery. In this scheme, you see function of immunoglobulin E. You see line of the epithelial cells. You see brown star-like structure of antigen. When this antigen enters the tissues of patients, of course, antigen becomes engulfed as any antigen by the antigen-presenting cell, for example, dendritic yellow cell shown here. Antigenic determinants of this allergen are present in the complexes MHC2 molecules, presented to T helper cells. These T helper cells, after activation, becomes mature from naive T helper 0 cells to the T helper cells type 2. These T helper cells type 2 produce interleukin 4, 13, interleukin 5, and these interleukins activate B cells, of course, such B cells that have membrane immunoglobulin D or immunoglobulin M receptors that specifically react to this allergen. Clonal selection is happening. B lymphocytes replicate, form the clones of the cells producing antibodies, and in the environment, in the lymphoid organ, where prevalent cytokines are interleukin 4, interleukin 10, interleukin 5, interleukin 13, or these interleukins produced by T helper type 2 cells. But in these conditions, immunoglobulin synthesis is switched from the immunoglobulin M to the immunoglobulin E. This immunoglobulin E, through the bloodstream, is delivered to the basophils, plasma cells, and eosinophils, attached to the Fc receptors, and when allergen repeatedly enters to this organism, they cross-link these immunoglobulins on the surface of the plasma cells. Plasma cells release histamine. A histamine attracts eosinophil to this area, participate in the recruiting of the eosinophils because they migrate through the vessels to this inflamed area, accumulate in this area, produce their cytokines, the mediators of inflammation, and maintain inflammatory reaction. This inflammatory reaction is named anaphylaxis. And very quickly, let's repeat what is happening in the process of the humoral immune response development and cellular immune response development. For repeating, antibody synthesis is based on the interaction of the at least 3 types of cells: antigen-presenting cells, what is this one, helper cells, this one, and B lymphocytes. Antigen-presenting cells engulf pathogen, destroy pathogen, isolate from these antigenic determinants, and present these determinants together with MHC2 molecule to the B cells, to T cells, T helper cells. T helper cells, via recognizing of antigen by T cell receptors, becomes activated and mature to the T helper type 2. T helper type 2 release interleukin 4 and 5. They stimulate B cells, but not all. B cells that before this are selected by antigen that specifically react to the immunoglobulin receptors of B cells. It means receptor must be specific for this antigen. And additionally, this B cell establishes a co-receptor contact of CD40, co-receptor B cell to the CD40 ligand of the T helper cell. Third signal is cytokines. It means you see three pathways of the activation of B cells. First, direct activation by antigen. Second, co-stimulation. And third, cytokine activation. You know that some antigens are named T-independent antigens. Usually they're polysaccharides with a high molecular weight. These polysaccharides, microbial capsule polysaccharides, may directly bind to the antibody receptors of B lymphocytes and activate these B cells for transformation of plasma cells and producing of immunoglobulin M. It means some polysaccharides may, without T cells, cause transformation of B cells to the plasma cells and synthesis of immunoglobulin M. But protein, lipid, other complex antigens of the carbohydrates to proteins, they usually cause T-dependent immune responses. It means responses where key role is role of the T helper cells. Usually antigen-presenting cells engulf and present antigens, well, this is a yellow, this MHC2 molecule, antigenic peptides to the T helper cells. T helper cells, when are specific to these peptides, becomes activated. Activated was this T helper cell, shown by the red membrane, releases after contact to the antigen presenting cells cytokines. You see the interleukin 4 and 5, the T helper 2 cytokines, and they stimulate activation of B cells by the antigens, replication of these cells, maturation to the plasma cells that after that produce first immunoglobulin M after G, but this antibody synthesis may be switched to the synthesis of the immunoglobulin A and immunoglobulin E also. You know that cytokines play critically important role in this process of antibody synthesis initiating. So, what the cytokines stimulate development of the antibody synthesis? They are cytokines of the second type, or T helper type 2. Molecules produce interleukin 4, 13, 10, 5. No, for example, interleukin 4 is produced by T helper cells type 2, mast cells, eosinophils, and basophils. It means cells that are stimulated by interleukin 4. What is happened? Interleukin 4 is anti-inflammatory cytokine that inhibits processes of inflammation stimulated by T helper type 1 cytokines, such as the tumor necrosis factor and interferon gamma. They are pro-inflammatory. Interleukin 4, oppositely anti-inflammatory cytokines. Interleukin 4 induces differentiation of naive T helper cells, Th0, to T helpers type 2. This is a type that of T helper that is responsible for secretion of interleukin 4. It induces B cell differentiation into plasma cells and B cells classes switching to immunoglobulin E. What we told about this. It means prevalence from the interleukin 4 stimulates switching of antibody synthesis to immunoglobulin E. Interleukin 4 decreases proliferation of T helpers type 1 and serves as the inhibitor of the interferon gamma that stimulates differentiation of naive cells to the helpers type 1. And interleukin 4 correspondingly cause the inhibition of the synthesis of interferon gamma, this is the helper type 1 product, and interleukin 12 by dendritic cells. What role of the cytokines? Steps of antibody production. First, binding of antigen, green, to B cells, or binding of antigen to antigen-presenting cells of the macrophage type. After that, processing and presentation of the antigen to T helper cells. What you see here, that B cell via combine of the antigenic peptide to the MHC2 molecule presents antigen, presents to the T helper cell type 2. With a parallel line, antigen presented by dendritic cells and macrophages may be presented to the T helpers precursors of T helpers type 1. Between these helpers occur struggling for what's enhancing of own clone. Third, direct delivery of CD4 T helper cells derived cytokines, interleukin 4 and 5. What you see here, that T helper type 2, after stimulation by the antigen presentation of antigen, stimulation of interleukin 2, stimulation of the presence of interleukin 4 and 5, they additionally stimulate B lymphocytes, what for replication, for transformation to the plasma cells and producing of the immunoglobulins of different types. Now you understand the dendritic cell, what presents antigenic peptide together with MHC2 to the TCR. A TCR needs co-receptor interaction of CD4 molecule to the MHC2, and this T helper 0 and TH0 naive cell may be activated only after that. No, well, switching is happening. We understand that in the prevalent presence of interleukin 4 and 5, immunoglobulin M and G producing switched for immunoglobulin E synthesis. By the way, the synthesis of immunoglobulin E is not shown, but we understand that this is happening. Now, here you see how B cell in the upper part, number one, serves as an antigen recognizing cell, because antigen binds to the immunoglobulin receptors. After that, the cell processes and presents this antigen together with MHC molecule type 2. This molecule MHC2 type 2 is presented to the naive T cell after recognizing of MHC2 molecule by CD4 receptor and co-stimulation by the CD40 receptor of the B cell binding to the CD40 ligand. This T helper cell is undergone to the replication and maturation to the T helper type 2 that produces interleukin 4 and 5. These cytokines bind to the corresponding receptor of B cell, and the B cell is transformed to the plasma cell. Helpers precipitating the humoral immune response are shown in the scheme by the dotted line. First of all, T helper type 2, via secretion of its cytokines interleukin 4, 5, and 13, 10, stimulate humoral immune response. T helper 9 and 17 also stimulate humoral immune response antibody synthesis, and T follicular helpers also, because T follicular helpers maintain replication of all the T cells, including T helper type 2. T helper type 1, they concur to the helper type 2 for the stimulation. And interleukin 4 serves as the inhibitor of synthesis interferon gamma and correspondingly of the transformation of naive T cells to the helper type 2. And correspondingly, T helper type 2, even interferon gamma synthesis prevents maturation of naive cells to the T helper type 2. It means T helper 1 and T helper 2 responses, they struggle opposite one to other. What the immune response would be developed depends on the presentation of antigen. If antigen, what shown red, is presented by the MHC2 molecule, what brown molecule contain two chains in the right part, this presentation is recognized by TCR of the helper cell, CD8 receptor to the helper cell after confirming that is a legitimate interaction. After binding to the MHC cell confirms that the helper cell and antigen presenting cell are cells of the same host organism. After that, signal of activation via CD3 molecule is produced and activates T helper for replication. If other presentation is happened, if antigen is presented with the complex OMHC1 protein, cytotoxic CD8 cell recognizes this antigen via T cell receptor, and CD8 receptor of this T killer cell confirms the legitimate character of recognition by contact of CD8 to the MHC1. And also signal of activation via CD3 molecule is transported to the nucleus of this helper killer cell. Now here you see how processing is happening. Antigenic peptide is combined to the matured of the MHC2 molecule, the process of maturation, and this molecule in the complex to antigenic peptide is exposed to the surface. On the surface, antigen is recognized by T cell receptor, CD4 molecule confirms that MHC2 molecule is own molecule. Of course, T helper cell activation will stimulate replication of the cell at maturation. Maturation may cause formation of the either T helper type 1 cells are the T helper type 2 cells, and polarization of T helpers has happened. Polarization into Th1 or Th2 population. This polarization depends on the presentation of antigen and the cytokines. If antigen is presented external exogenous antigen of bacterium engulfed, if this peptide is combined to the MHC2 molecule and present in this complex, humoral immune response will be stimulated. If the antigen as the part of the endogenous parasite, for example, the C virus, would be ruptured for the small peptide fragments and injected to the endoplasmic reticulum, and what in this reticulum will be combined to the MHC1 molecule in the process of maturation. In this case, in the complex with the MHC1 molecule, antigen would be exposed to the CD8 T killer cells with the cellular immune response. Cell immune response, of course, is mediated by T helpers type 1. These T helpers secrete interferon gamma, maintain activation of cytotoxic lymphocytes, and interleukin 2 that maintains replication of all the cells, including cytotoxic T lymphocytes. They attack and kill target cells infected by the viruses. You see here how CD8 cell with the granules contain granzymes and perforins, recognized by T cell receptor, antigenic peptide presented by MHC1 molecule on the surface of target cell, this virus-infected cell. And T killer cell needs confirmation of the legitimate character of this interaction by CD8 molecule that will react to the correctly react to the MHC1 molecule of the antigen MHC complex. After that, T killer cell will release content of these granules to the space between T killer cell and target cell. Perforins, shown green, will form holes in the membranes of the target cell, and enzymes will be injected inside, causing destroying hydrolytic destroying of nucleic acids and proteins. Here you see gamma delta T cell receptor, what green on the surface of gamma delta T cell. This is a killer cell. Why killer? Because it recognizes what this green microbial antigen, the complex to the MHC1 molecule, with this molecule. And these gamma delta T cells serve a very important role in the elimination of dangerous own cells and tumor own cells and virus-infected own cells. Now here you see how orange cytotoxic T lymphocyte recognizes microbial antigen in the complex of antigenic peptide and orange MHC1 molecule. TCR recognizes this antigen, CD8 recognizes their own this MHC molecule that forms the signal of degranulation for cytotoxic cell. A cytotoxic cell shoots the target cell by release of the granzymes and perforins. And here you see this process, what has happened. Exception for granzymes and perforins, cytotoxic cells may kill target cell by initiation of the apoptosis in the cells. Apoptosis is initiated by binding of Fas ligand of cytotoxic CD8 lymphocyte to the Fas receptors of the target cell. Fas ligand is capable of reacting simultaneously to many, at least 3 Fas receptors. These 3 Fas receptors form the complex. Their transmembrane portions combine in the cytoplasm cell, stimulating procaspase synthesis. Procaspases initiate signal that finally causes formation of the active caspase A that attacks nucleus, attacks DNA, causing destroying of the target cell. Now here you see figure demonstrating accumulation, initially lowering of concentration of different immune cells in the process of immune response. Initially, you see that antigen-presenting cell stimulates B cells and T cells. No, B cells are stimulated without antigen-presenting cell, directly by antigen that may bind to the immunoglobulin receptor of these cells. T cells need to be activated by antigen-presenting cells. After activation, both B and T cells, T helper cells and T killer cells, then they go to the proliferation. Their descendants colonize many immune organs, and finally B lymphocytes mostly are mature to the plasma cell that produce antibodies. These antibodies usually eliminate microbes. These cells also participate in the elimination of the antigen of the infected, so virus or bacterial infected own cells. Finally, antigens disappear. In the absence of these antigens, majority of the cells of clones of the activated T and B cells that are specific to these eliminated antigens undergo to that apoptosis, and only memory B T cells wait for the next episode of the contact with the antigen to generate secondary immune response. This is the last slide. Lecture is finished. Thank you for your attention. Goodbye. Алло. Димочка, я закончил чтение лекции, и у меня к вам просьба, вы можете ее тоже скинуть мне на компьютер, чтобы я разместил на сайте кафедры эту лекцию? Ну, конечно, конечно, Дим, это не проблема. Я потом просто буду группу вести, ну вот, скинете очень хорошо. Спасибо. Спасибо вам, спасибо. Спасибо. До свидания.